my pleasure to introduce Mark Goldman. He's faculty at UC Davis. Mark did his undergrad at Stanford, then his graduate work at Harvard, and his postdoc at MIT. He has done a bunch of very nice theoretical and computational neuroscience work in a variety of neural circuits and systems that we'll hear about today. But he's also an HHMI professor at Davis, where he's also had a Sloan Fellowship, and I think the award that maybe we should most celebrate is the Outstanding Graduate Mentorship Award, especially for this environment. I would like to highlight that. So Mark, please take it away. Thank you for coming.
Great. Well, thank you very much for inviting me, and I've already had some great conversations this morning. Looking forward to more. You really have an amazing department, and it's amazing how many people were on the list, and then how many people I'm not even going to get a chance to meet with who I would love to. So today I'm going to talk about some efforts to tease apart microcircuits of the brain, and both have an element of some general modeling issues, even if you care nothing about the microcircuits I'm going to talk about, which are mostly going to be in the eye movement system, as well as, I think, some more general conclusions about some problems that are fairly fundamental to thinking about brain computations. And this is where I'll start. So the first half of the talk, I want to talk about a really simple to pose, but it turns out very difficult to answer problem. And it's the following. So in many memory and decision-making circuits, this is a cartoon, we see neurons who accumulate and maintain signals on the 1 to 10 second time scale. So what you see here in the cartoon is a stimulus which is presented transiently. This is my cartoon of neuronal activity whose firing rates accumulate in the mathematical sense, that is, mathematically integrate that signal. And then in the absence of the stimulus, that activity is maintained. And of course, this is the essence of short-term memory, to have the neuronal firing rates maintained in the absence of a stimulus. And I should comment that everything I'll talk about is actually analog short-term memory, in the sense that if the stimulus were larger or wider, then the accumulation would have gone up to a different level, and the circuits I'll be talking about all can maintain that analog level of activity. So where's the puzzle here? So I mentioned the time scale of 1 to 10 seconds. Well, that's much longer than the classic time scales we think of for single neuron dynamics. So, our classical sort of model of an integrate and fire neuron would be that a stimulus comes in, the firing rate goes up, and then when the stimulus turns off, there's a decay of activity on some biophysical time scale. I'm just calling it tau neuron. It might be in the neuron, it might be in the synapse, of 10 to 100 milliseconds. And the circuits that I'll be showing you, at least intracellular current injections suggest more like these time scales for the decay of currents. So the question is, how do systems bridge these time scales and develop the long time scales of short-term memory? And can we figure this out in a real circuit? So the question today will be really focusing at the circuit level. What circuit architectures or motifs generate such persistent activity beyond the time scales of the intrinsic neurons? So let me just start you with the standard toy model of how these systems work. So the standard toy model is a command input comes into the neuron shown here. The firing rate goes up, and then it decays on this neuronal time scale. Well, and typically, again, this is a short time scale, but neurons aren't in isolation. So imagine that this neuron is part of a circuit which has self, and think of this really as probably a population of neurons with self-excitation and then mutual inhibition between these two neurons. Well, in that case, the story can be the input comes in, it activates these neurons, and then through either the self-excitatory connections or through a double negative form of positive feedback, these neurons can maintain their activity. So basically, they start firing due to the command input, and then they excite themselves, or they inhibit the neurons that were inhibiting themselves. And in that case, activity can be maintained over long time scales. So this is the basic idea. This is actually from a paper looking at a monkey prefrontal persistent activity experiment, but I'm going to actually switch to showing you a model system for this. And what I want to point out, again, because you'll see it in the model system, is the two different possible pathways for positive feedback. One shown here, which is recurrent excitation, and the second one being this double negative form of recurrent disinhibition, where this population inhibits the population that we're inhibiting it. Okay, so now switching to a model system, I actually want to show you a similar circuit that exists in the eye movement system of vertebrate animals, and I'll happen to show you experiments from goldfish. In this talk, you can ask me about some later zebrafish work that we've been doing, and I should say we refers to these data are from David Tank and Emery Oxai, my experimental collaborators. I'm the theory part. Okay, so here's the, so here's the behavior, and hopefully this will play. So you're going to see a zoom in on the fish's eye, and again, the fish's eye will move very much like your eyes or my eyes move as well. So this is the typical way we move our eyes when our head is still and there's nothing in the world that's smoothly moving to track. We don't move our eyes smoothly. We instead have fixations and rapid saccades. That's the rapid eye movements between the different fixations. And the question here is going to be, how does the fish keep its eyes still? Because the muscles of the fish eye would tend to make its eyes relax back to a central position. So it needs persistent neural commands to hold those eyes still at a given position. So here's a cartoon picture of that. So, and these are actual neural recordings. So this is a neural recording of eye position in the fish, and the fish you can see here is making a nice set of saccades and fixation, saccade, fixation, and so on. So in terms of the circuitry, these rapid saccades are driven by eye velocity coding command neurons. They weren't recorded in what I'm showing you, so I'm just showing you a cartoon. But there are excitatory commands, brief bursts of neural activity that drive the eyes one way, and then inhibitory neurons that burst and drive the neurons, drive the eyes the other way. And again, what I was trying to convey previously was that if the eye musculature just received a brief command, the eyes would move, but then because of the spring-like muscles, the eyes would move back to a central position, and they wouldn't be maintained. And this immediately suggested that what one needs to maintain the eyes at a given position, it's really Hooke's law, they need a position command. And that immediately suggested there must be a mathematical integral from velocity coding command neurons to a position coding command neurons, and that's done by a devoted brain circuit called the oculomotor neural integrator. And you can see the recording of one of the neurons in that brain circuit here. So a pulse comes in, the firing rate goes up, and it persistently maintains an elevated firing rate. Another pulse comes in, and it persistently maintains a higher firing rate. So in this case, there is persistent activity that stores a running total of the input commands. And this is the memory that I was referring to earlier. So this is really a memory of the previous set of eye movement commands. In this case, it's an integrator, so what it's storing in memory is a running total. And again, you can see that it will, when there's an inhibitory pulse, it will step down. Okay. So in general, I won't be showing you raw traces. I'll be focusing on tuning curves. So I'll just summarize this data by the firing rate as a function of eye position during one of these fixations. I should note all the recordings are done in the dark, so there's no visual feedback. So again, this is maintained without visual feedback. And each of the dots in this graph refers to one fixation, what the firing rate during that fixation was. And you can see this nice threshold linear relationship between firing rate and eye position. Just to show you what these data really look like, this is actually a movie of the eyes, and unfortunately I don't have the audio plugged in right now. Anyway, you would be able to hear the action potentials crackling. That's okay, but what you're going to see is every time the animal's eye moves, you can see basically this is tracking the eye position and the corresponding firing rate. And you can see the firing rate being maintained during a fixation. You see that the saccade came in, that's the deviation from that line. Saccade came in, and you can see the firing rate tracking the eye position and holding an eye position along this diagonal red line here. Again, that's the tuning curve of this neuron. Okay, so that was just showing you one neuron. Really, there's a large population of neurons. So that was one neuron recorded on the right side of the brain. If you looked at all of the neurons, you could look at their firing rates as a function of eye position as the animal holds different eye positions from left to right. And what you see is these neurons all have these threshold linear relationships, but there's different neurons have different slopes and different neurons of their relationship, and they have different thresholds. And when you get into the details, I'll refer to neurons as having a low threshold if they recruit quickly as the eyes move from one side to another, and having a high threshold. So these would be high threshold neurons, and these would be low threshold neurons. And in motor systems, this is very, very common to have different thresholds and recruitment patterns. And I should say then there's also neurons on the opposite side of the opposite side, and statistically they are a mirror image. In fact, in this case, I literally just created a mirror image, but because actually the data set we have has some from the right and some from the left, and we just pretend that there's one on each side, but statistically they're a mirror image. The nice thing about the fish system is that these neurons with the increasing slope as the eyes move from left to right all appear on the right side of the midline of the brain, and those with the decreasing slope all appear on the left side of the brain. So it's beautifully organized. There's two populations on each side, an excitatory population shown in green and an inhibitory population shown in red. And we believe that the gross architecture is primarily as follows, that the excitatory neurons project ipsilaterally, that means to the same side, and the inhibitory neurons project contralaterally, that means to the opposite side of the midline. And what does that mean functionally in terms of the feedback loops that I was talking about earlier? What that means is here we have the recurrent excitatory loops that I was mentioning sit in all of the generic models of persistent activity, of memory storing activity. And we also have the double negative form of positive feedback, because neurons on this side of the brain, they have to get to the other side of the brain, they have to go through an inhibitory neuron, and then to get back, they need to go through another inhibitory neuron. So basically you can play this game. If you want to get from any neuron back to itself, it'll either go through excitatory neurons or through even numbers of inhibitory connections, making the double negatives. Okay, so we wanted to build a model of this, and here's our model. It's actually going to be a spiking model, but it's a lot easier to understand it just as a rate model, so I'll explain the rate version of the model. We basically broke each of these populations into 25 neurons each, because we think in the fish there's about 100 neurons participating in this circuit, and what we want to fundamentally model is how the firing rate changes. What drives a change DRDT in the firing rate? And you can say in the absence of input, there's certain dynamics of leak. That's actually characterized experimentally and captured by a function f of r, a function of the firing rate. There's same-side excitation, and that's driven through we're modeling that as a sum of inputs from all of the neighbors. So this is neuron I. It receives input from neurons J, and it goes through, we assume, some synaptic or dendritic nonlinearity S. And so this is S of R. So basically neuron J projects to this neuron, goes through a nonlinearity that is then weighted by a weight. You sum up all of the inputs, that's a total excitation. And the nice thing is the excitation comes from the same side. Likewise for inhibition, so there's a different set of weights and a different nonlinearity in general for inhibition. And then there's also some tonic background inputs and burst commands. Those are actually the saccadic commands that move the eyes from one place to another. So our question is, well, what if you want to have persistent activity? Persistent activity is when DRDT equals 0, and I should say persistent activity when the burst command isn't there during a fixation. So that basically says for persistent activity, these terms must all sum to 0. Beautifully, if those sum to 0, we get more than persistent activity. We actually get the mathematical integration. We get that if DRDT is proportional to the burst command input, then R is proportional to the integral of the burst commands. And this is how we fundamentally think integrators come about. How, and whether it's accumulation of evidence in a cognitive task, or whether in this case it's a velocity to position integration. So basically it's the same mechanism that causes the persistent activity and the integration, and what it really is a balancing of the intrinsic leak being which would tend to make the firing rates decay, being offset by the synaptic inputs. Okay. So I'm giving a reference here for fitting methods. So I'm going to give you an essence of how we fit these models to actual data. So basically we build a conductance-based spiking model by constructing a cost function that simultaneously enforced a whole set of experiments. Our goal was basically to take a whole suite of experiments that had been done over a decade on this system, put them all together into one cost function that says the model needs to try to match each of those experiments. So there was an intracellular current injection experiment, which was basically trying to get the intrinsic responses of neurons, f of r, and how they respond, how at the somatic level they respond to input currents. Then we had a database of single neuron tuning curves that gives us the firing rate as a function of any given eye position the animal is holding. And then we also, and I'll show you those in a moment, have firing rate drift patterns following focal lesions. And we basically make a different cost function term, which is, for the experts, just a quadratic squared error type of term that enforces each of these experiments. The unknowns is really the unknowns in most neuroscience experiments. Very rarely do we know what the actual synaptic weights in a circuit are or the weights coming from external inputs. So that's where we hope the model can help us say something about these. And also, we don't know the form of the synaptic or dendritic nonlinearities. So again, what we're going to do is actually, it turns out, assume a form of synaptic nonlinearity, chunk through and pull out through a regression procedure what the weights are, and then try different synaptic nonlinearities. So we basically have a two-parameter function for the synaptic nonlinearities that can be saturating, it can be sigmoidal, it can be superlinear, it can be linear. Okay, so here's what comes out when you do this. So this is an example of one of, again, this was actually a spiking conductance-based model. So here's the model. You can see the saccadic inputs are coming in here. It causes a brief burst of firing, but then what I want you to focus on is during this fixation period, here you see this nice persistent activity. There's another saccadic input coming in that gets integrated, and now you see higher rate persistent activity. So this is the integration. Usually we'll just plot firing rates, so that's what's being shown here. Green is just to guide your eye of what a perfect integral of the saccadic inputs would be. Gray was actually the raw model, and black is what you'd see in a paper with after putting a little bit of smoothing on it. And what you see, this is the firing rate of one neuron as a function of time, is basically its firing rate is perfectly following the integral, as the animal would be in each of these vertical pieces is where a saccade came in the model, and then the flat pieces are where the fixation was. So just to summarize that data, this is showing you for 4 neurons, but it worked for every single neuron. It's a little bit backwards from what you might think. The solid line is actually the tuning curve from the experimental database. And what you see in the boxes is actually the responses of the model at different eye positions. And we also were trying to fit some noise distributions, so that's why you see box plots. That's actually conveying the noise in the model. So here are 4 neurons. 2 of these were on the left side of the animal. 2 were on the right side. One is what we'd call a low threat. The blue and the green are what we'd call low threshold neurons, and the red and the black are high threshold neurons. So the main point is, first, we have a whole circuit where we have a large number of tuning curves, and this is one of the very few models in a vertebrate system where we actually have a model that recapitulates the data at a neuron by neuron level. So we can make a one-to-one mapping between neurons in the model and neurons in the system. So we were happy with that, but what do we learn about the system from this? And this was really the key experiment that I think provided the most insight. So it's the following. So my experimental collaborator, Emory Oxai, he inactivated the left side of the circuit and recorded from neurons in the right side of the circuit. And now you need to remember a little bit about the circuit, that the crossing connections between the two sides are inhibitory. So what I argued was, to get persistent activity, you needed this balance of currents to get persistent activity. In some sense, we've now undone that balance because we've lost inhibition. So naively, what you'd expect is firing rates would start drifting upwards, because you've unbalanced things to the point of having not enough inhibition. And that's indeed what you saw in the recorded neurons at low firing rates. You see very sharp upward drift in the firing rates seen here. The mystery and the surprise was that at high firing rates, the neurons remained stable. Their activities didn't drift upwards. And we could recapitulate that in the model. You see again here the model sharp upwards drift at low rates and persistent activity at high rates. We could recapitulate that if there was some sort of threshold. So let me just summarize. Persistence was maintained at high firing rates experimentally. These high firing rates, we now need to think a little bit about those tuning curves. So when these neurons on the right side are at high firing rates, remember one side of the brain's tuning curves increased as the eyes move from left to right, the other side decreased as the eyes move from left to right. So that means when these neurons are at high firing rates, these neurons were at low firing rates. So the high firing rates occur when the inactivated side would have been firing at low rates. So what this suggests is that these low rates were below a threshold for contributing. Effectively, what the network is doing is it's really the high firing rate neurons are driving the network, and the low firing rate neurons are below a threshold for influencing the circuit. So I've left out a longer story. We actually have some, out of this came two possible mechanisms for such a threshold. One was actually the actual firing threshold, sort of a neuronal threshold, was one possibility, and the other possibility was that there's a synaptic threshold, and biophysically that could be a synaptic facilitation, where low rates basically don't facilitate the synapses and cause much transmission. The other thing on excitatory synapses could be something like an NMDA spike, and there's known to be a lot of NMDA on the excitatory synapses here, so it may be that low firing rates aren't enough to trigger an NMDA spike or an NMDA plateau. And again, you can look at the work this is published if you want to get into the biophysical implications. I want to actually step back and just talk about macroscopically what this says about the macroscopic mechanisms for maintaining memory in this circuit. Because I presented to you two possible mechanisms by which persistent activity could be maintained. One was recurrent excitation, and the other was recurrent disinhibition, being a double negative form of positive feedback loop. So now let's just kind of reason through this. Look at the network activity when the eyes are directed rightward. So when the eyes are directed rightward, the right side neurons are at high firing rates, and there's a positive feedback loop here due to recurrent excitation of these neurons exciting each other. But now let's think about the inhibitory feedback loop. The left side neurons are at low firing rates, and we just said they appear to be below threshold for transmitting. So that means we've broken the loop. So the right side would be transmitting inhibition to the left side, but the left side, and that's why I'm showing dashed lines here, is below threshold for transmitting back to the right side. So due to thresholds, there's no mutual inhibitory feedback loop. And I think in the era of connectomics, this is really, really important, because this is a case where the old theories of how the circuit worked all said that the primary positive feedback loop should be disinhibition. It was purely on theoretical grounds, so it wasn't that they had empirical evidence, but they just thought, gee, well, here's this double negative loop, we think that's how it works. But here we have a case where functionally there's really one way, unidirectional inhibition from the high rate side to the low rate side. And then when the animal moves its eyes to the other side of the head and the left side becomes active, then the inhibition goes from the left side to the right side. And that belies the fact that anatomically it looks like there is a double negative feedback loop. So really there's no feedback loop here, and the physiology, because of thresholds, belies the anatomy. And so excitation, not inhibition, maintains the persistent activity, and inhibition is anatomically recurrent, but functionally feed-forward. Okay, so that's at really just a gross level of saying what are the key contributors to positive feedback and maintaining persistent activity and integration in the circuit. I now want to delve down to what about the microcircuitry? And here's the problem I want to address. So I said we fit a weight matrix, but even in a 100 neuron circuit, there are 100 squared potential connections, 10,000. Turns out that because excitation only goes to the right side and inhibition to the left side, we're down to 5,000. Only half of them are possible. But how do you even say anything about what the connectivity is? I mean, it's like the worst statistics problem ever. You're trying to figure out 5,000 parameters underlying this behavior. So what I want to do, and it really follows on a history of work in what's called sloppy models coming from the physics literature, is say how one can work through this problem and try to figure out at least what is important and maybe what isn't important. I hope this is a generally valuable lesson for whatever system you work on. So here's my cartoon of the model cost function. Remember, we had a cost function for fitting the data. And here I'm just going to make it really, really simple. Imagine that we just look at two individual synaptic weights. Oops, what did I just do? Sorry. Two individual synaptic weights, W1 and W2. In this case, W1, it's really sensitive. If that's tuned correctly, you get the right model behavior, and if it's mistuned, it's going to cause a lot of cost and your model's not going to fit. On the other hand, W2, this synapse really, it doesn't matter what you tuned it to. You're basically moving along the trough of this cost surface, and that's a very insensitive direction. Now, in general, what it will be is not individual synapses, but in fact, linear combinations of synaptic weights that the model fitting is sensitive to or insensitive to. So what do? The cost function curvature is described by its matrix of second derivatives, the Hessian matrix, d squared cost, dwi, dwj. And what you can do is to figure out where the steepest and the least steep directions are is you just do principal components analysis on this matrix. And that will identify the patterns of weight changes to which the system is most sensitive. And when you do this, what we found was there were only 4 most sensitive components. Those are the ones that jumped out in the principal component analysis and corresponded to the directions of steepest curvature. And the others were largely insensitive. So I want to show you, I'll show you the first 3 of these. And excuse the crowded slide, I'll work you through it. So these are the principal component 1, 2, and 3. And what I'm plotting here is the components of the principal eigenvector. So each neuron in this model received input from 50 other neurons. The first 25 are its excitatory inputs from the same side. The next 25 are its inhibitory inputs from the other side. Here I happen to order the inputs from the lowest threshold neurons to the highest threshold neurons. But for this first principal component, you didn't need to know much. Not surprisingly, the most sensitive thing you can do to screw up the model is to make all the connections more excitatory. So the direction positive here either means excitatory connections getting more excitatory or inhibitory connections getting less inhibitory. So hence effectively more excitatory. If you make a perturbation of a fixed vector length along this direction, it's a complete disaster. The neurons just go skyrocketing until they saturate because you've made everything more excitatory. So the next most important, and I should say, eigenvectors only indicate a direction, so you could also equivalently make everything less excitatory. So I just had to pick one for illustration. So the next component would be either strengthening or weakening all of the synapses, excitatory and inhibitory. So here I'm going to show you making the excitatory connections less excitatory and the inhibitory neurons less inhibitory. More excitatory, less inhibitory. So I'm just turning down the absolute value of the weights and everything. You do that perturbation and you get just an exponential decay to an intermediate fixed point. So you can see everything's decaying to a fixed point around here. The next component I won't get into has to do with the balance of low and high threshold neurons and so on and so forth. What I want to show you here is eigenvector component 10. Here you can see that there are groups of neurons that are either made more excitatory or less excitatory. But look what happens when you make the same magnitude perturbation of this pattern to the neurons. Basically nothing. You still have a beautiful integration where you see saccade-driven steps and maintaining persistent activity during the intervening intervals. This is showing neurons, and I'm only showing you one neuron, but this works across all the neurons. So now you can actually step back and say, well, let's push this idea of insensitive directions further. So here I'm actually showing you the weight matrix for one of the models. So again, I've broken down the neurons the same into 100 neurons. The first 50 are the left side, the second 50 are the right side. The first 25 were the excitatory neurons, the second 25 are the inhibitory neurons, then excitatory neurons, inhibitory neurons. So for example, this block would be the left side excitatory to excitatory connections. And here I've actually, sort of anticipating some work we've done in the zebrafish, actually given them spatial locations. So here I ordered them from rostral to caudal. So this would be a fairly all-to-all connectivity pattern. And here's another circuit where there's what I'd call a topographic pattern. So you can see the strength is mostly along the diagonal, so those are very local, spatially local connectivity. Now here's your psychophysics test. These are the outputs. This is the firing rate of one neuron, but I could have shown it to you for any from the left side or the right side. Again, the bright neon colors are just to guide your eyes of what a perfect integral is. Black is actually showing, this is the average of the right side, but I could have shown you individual neurons too. They're very similar. This is the average of the right side activity in black, the average of the left side activity in gray. I mean, they are just identical. I mean, again, I would love to do psychophysics on having someone distinguish between these, yet completely different circuit architecture. One all-to-all excitatory connectivity, one local excitatory connectivity. So is there a formal way we can see that in some sense these circuits were identical in what was important, in the sensitive directions? Well, what we can do is we can take these weight matrices, these patterns of activity, and project them onto the sensitive and insensitive eigenvectors of the cost function. And that's what we've done here. So what I'm showing you is in log units, so these are exponentially different. When you see 10, that's e to the 10. And here's the eigenvector component. So eigenvector component 1 was the most sensitive one. Eigenvectors, higher numbers, are insensitive ones. And what you should see, remember I told you that there were 4 sensitive components? Well, this is now what I didn't say something. I took the difference between these two weight matrices to say where are they different, and projected the difference between these two weight matrices upon the sensitive and insensitive eigenvectors. And what you see is that the difference between the two matrices is only really, the differences are nearly 0. So this is like e to the minus 10 difference between them. Essentially 0 in the 4 sensitive components. So from the point of view of the sensitive components, these matrices are identical. And all of their differences are in these insensitive components. And I should have made clear, the insensitive components are basically components where you have excitation and inhibition that are canceling each other. So either you're getting more excitation from one cell and less excitation from another, or you're getting more excitation from one cell and more inhibition from another cell, so that the net change in input is 0. So hopefully we think that this is the way we're going to have to look at circuits. We're not going to know exactly what the connectivity is, but hopefully we can figure out what are the important features of the connectivity for making the circuit work, and what really is driving the circuit, and how do we say what's sensitive and what's insensitive, what's important, what's not.
So when you do this sensitivity analysis, you have to pick some weight configuration in order to evaluate the Hessian.
Right. So this one, because we did a quadratic cost function, it turns out the Hessian is identical everywhere in the space. So that was a upfront modeling trick that we did because we knew where we were going and we knew what we wanted to get out of it. So the Hessian here actually is everywhere. There are some subtleties because of signs that you actually have to look at the gradient. If you have synapses which are truly excitatory or truly inhibitory, weights can't flip from negative to positive. So you do have to look at the components of the gradient when things are on the boundary. It turns out the directions of the gradient line up basically cover the same space as those first 4 eigenvectors. So there's nothing new really to learn from those. But yes, good question, and kind of expected to answer that, so thank you for asking it. Okay, so summary of this part. I briefly mentioned neural integrators are circuits that accumulate and store a running total of their inputs. They're important both in motor control, as you saw here, as well as in more cognitive tasks like accumulation of evidence for decision making. A model system for those is the oculomotor neural integrator. And there, positive feedback appears to be due to excitation, not mutual inhibition, and that's what appears to be critical to memory storage. The modeling issue I brought up, and which I'm going to continue into the second half of the talk, is this degeneracy of model fitting solutions. And I think this is just inherent when we talk about something like fitting the weights of a connectivity matrix. And one key question is, does this model degeneracy reflect lack of experimental constraints or patterns of connectivity that may differ from animal to animal? In a given animal, at a given time, there is a connectivity matrix. But I think this brings up the question, even if we measured the exact connectivity matrix in one animal, which we're actually trying to do, how important would that be versus still needing to figure out what was important about that connectivity matrix and what wasn't important about it? And I'm guessing if you look from animal to animal, what's going to be preserved is the sensitive directions, and what's not going to be preserved is the insensitive directions. And if you think about reinforcement learning would naturally pick up and try to correct the sensitive directions, but would basically let go free without any punishment in sensitive directions, because there'd be no feedback to the animal that those had somehow diffused to other parts of the parameter space, if you wish. Okay, part 2, different background color, sorry about that. Model degeneracy. Now I'm going to step to plasticity and show you how some of the same nasty issues can come up, and I think also push an interesting debate from the field of cerebellar plasticity. So model degeneracy and identifying sites and signs of plasticity. So what I now want to talk about is another eye movement. It's called the vestibulo-ocular reflex, and basically the punchline is vestibular input that comes from head turns can also drive eye movements. So here's a video basically showing an animal doing the VOR, the vestibulo-ocular reflex. And this is all based on monkey experiments, so that's supposed to be a monkey. So this is a reflex you have. It's your gaze stabilization reflex. When you turn your head, automatically through a reflex, your eyes will counter-rotate, and that's how you keep images in the world stable to your head movements. So the sensory input here was this back and forth sinusoidal head velocity. And again, what you see here is the motor output counteracting that. And I should say that the gain of the VOR reflex is the ratio of the eye velocity to the head velocity. So I'm going to use that a lot. I'm going to talk about the gain being the ratio of eye velocity to head velocity. A perfect gain for a normal animal would be 1. For every degree of head movement, you move your eyes 1 degree to offset that head movement and keep your eyes in looking the same direction. So here's the nice thing about this system. Even though it's a simple reflex, you can train the gain to go up and down. You can fool animals into adjusting their VOR. So this is how you do gain up training. So train the VOR so that you turn your eyes more. What you're going to do, and you're going to see it right here, is you have the visual image move oppositely to the head. So now you see that the monkey needs to move its eyes more in order to keep its focus on that banana. Likewise, gain down. So now we basically tell the animal, hey, you shouldn't counter-rotate your eyes. You move the banana with the monkey's head, and now the right thing to do to stabilize vision is not to move your eyes at all. So what happens if you train with this gain up or this gain down paradigm for long periods of time? Oops, sorry. Okay, so this is for gain up. What you see is here's the head velocity, here was the eye velocity before training, and then after training, what you see is that the VOR gain goes up, the eye velocity output will be increased. And the testing here is in the dark, so just moving an animal back and forth in the dark and you see the gain goes up. Likewise for the gain down case, the VOR gain, which started out again at that level, decreases. Okay, so those are the basic paradigms, and now I want to talk about the circuitry underlying this. So here's the basic circuitry. Again, the vestibulo-ocular reflex is a very simple reflex, and there's a direct reflex pathway. I'm simplifying it a little bit, but to connect to the previous side of the talk, your input is head rotation. Basically, from your ear canals come velocity commands telling you what your head velocity is. That goes through the same neural integrator that I was telling you about earlier to send out position commands that then lead to pulling the muscles and pulling the eye muscles and making the eye rotation. So that's the basic direct pathway. But in terms of adjusting this reflex, it's known that if you get rid of the cerebellum, you can't adjust this reflex. So there's also a side pathway that goes through the cerebellum, goes into the granule cell. I'm making a cartoon, and the output of the cerebellum are the Purkinje cells, and they provide actually inhibitory output to this basic circuit for the reflex, the basic direct circuit for the reflex. So the question I want to ask is, what plasticity mechanisms, specifically, what are the sites and signs of plasticity within this circuit that enable the velocity command to be increased or decreased? And here's the key issue, and again, hopefully stepping this back to really, if you work in learning and plasticity, to very basic issues that come up in kind of any system. So the key issue is identifying the sites of plasticity and how it's a challenge in neural circuits that have feedback loops. So here is a toy circuit, and we have an upstream neuron and a downstream neuron. I kept the signs. The Purkinje cells are inhibitory, and there was a minus sign. That's when you move your head one way, the eyes move the other way. But we don't really need to get into those details. Suppose you're doing a recording and you see that this neuron right here decreased its activity. If it's just a simple feed-forward circuit, you know exactly where the site of plasticity was. It was at this synapse or someplace further upstream from that synapse. So that's pretty easy. There was some functional depression of this synapse. And I should say for this talk, I'm going to refer, just because it's easy to remember, to LTD and LTP, long-term depression, long-term potentiation. I really don't know here whether it is, for example, depression of an excitatory input or potentiation of an inhibitory input. So I'm really talking about functional depression or functional potentiation of pathways. But anyway, in the feed-forward case, it's really easy. You see this firing rate go down, you know something upstream of it had depression. But now let's consider the trickier case where we add a feedback loop. So now we have a feedback loop from the motor command back to this neuron, and we see decreased activity. Well, here's another possibility. What if this synapse right here underwent LTP? That would increase the activity of this neuron, and through this inhibitory connection, then decrease the activity of that neuron. And then you have no idea what happened at this site. It could have been LTP or it could have been LTD. So this is the problem once you add a feedback loop with identifying sites and signs of plasticity. Well, it turns out that the cartoon I just gave you is the essence of a 35-year-old debate in the cerebellum between the Marr-Albus-Ito model of the cerebellum and the Miles-Lisberger model of the cerebellum. And you can find this, these names in Candel, so any basic neuroscience. But this is, I boiled down the essence of the argument. So let me just step through it really, really carefully. First, the physiology. So the physiology is when you do training to increase the amplitude of the VOR, to increase the gain of the VOR. You see that Purkinje cells, like I showed you in the previous cartoon, decrease their firing rates. The cerebellar nuclei firing rates increase. The eye velocity increases. Okay, and it's in amplitude. It's still going the opposite direction. So now the Marr-Albus-Ito theory says, oh, this is really simple to explain. There was LTD here. That decreased these cells' firing rate. They inhibit these cells, and that's what increased their firing rates. Nice and simple. So Miles and Lisberger come back, and they say, we're going to do some behavioral recordings. I'll show you what the essence of it was, but they did some behavioral recordings to try to isolate this pathway, this vestibular pathway. And they said, functionally, when we try to isolate it, it looks like there was LTP on this synapse. And if there's LTP on this synapse, they said that's backwards from what we would have expected. So there must have been, that wouldn't have made these firing rates go down. So it must have been that there was LTP down here in the direct pathway, and then due to an inhibitory synapse, that comes back up and causes decrease in firing there. And this debate has raged on for 35 years. The one thing that neither of these theories focus heavily on is that there's another feedback loop which they're not really paying attention to carefully, which is that eye velocity itself, what's the point of moving your eyes? It's to keep the world stable. Do they really keep the world stable? No, they're always slipping a little. So there's also a visual feedback pathway which comes into these Purkinje cells. And they haven't really considered that. So we set about trying to go back and figure out the fundamental question, is efferent's copy, this is called an efferent's copy of the motor command, just a copy that's sent back, is it necessary? Because that efferent's copy positive feedback loop here is fundamentally the difference between these theories. So Miles and Lisberger said, absolutely you need it. Our experiment says that there's LTP here, and therefore the only explanation has to be that the main plasticity driver was in the direct pathway. So what did we do? We built a simple model of this circuit. It's all based on linear filters. So there's basically, here's your different sites, the Purkinje cells, the brainstem neurons, and there's eye velocity input, and we just put linear filters that we're going to fit at each of these different sites. And we're going to attempt to fit the data, the key one is here, with and without this feedback pathway. And actually we do graded levels in between as well. What data do we fit this to? It's actually data from Jennifer Raymond's recordings in Steve Lisberger's lab. She had 23 different conditions, combinations of head movements and visual inputs at different frequencies. So for example, that the one which I showed you for increasing the gain or decreasing the gain, where either the visual stimulus is moving with the head or the visual stimulus is moving opposite the head. That would be a condition, for example. And after learning there were 15 conditions, mostly it was behavior, fewer neural recordings. Okay, so here's the surprising thing. So I'm just going to now show you, 4 of the 23 conditions. So this is going to be the stimulus. This one's one where only the head was moving in the dark, and this is one where vision was on and the head and the vision are the same. So this is where basically the animal's having the visual world move with its head. And then the same thing with steps rather than sinusoids. These are Purkinje cell recordings during that. These are eye velocity recordings during that simultaneously. So we try the no positive feedback model, and it fits remarkably well. Almost perfectly fits the eye velocity traces and the Purkinje cell traces. I mean, this is neural data, so fits remarkably well. And then you try the with positive feedback model, and it fits almost identically. So here's another complete degeneracy between these two models, despite the fact that each of these camps have been saying it has to be this one or it has to be the other one. And I should say that for the Mar-Albus-Ito model, most of the molecular studies suggest that there is depression at that site. So in some sense, we have 2 different cases where there is seemingly experimental evidence on each side of the debate. Okay, so we want to now look under the hood and say, well, okay, so they both can fit the data, but I'm guessing they're going to fit the data in different ways. So let's see how they fit the data. So first, the punchline is going to be the 2 models predict different patterns of weight changes during learning. First, I want to show you something that the models agree on. In both models, if we look at this brainstem site of plasticity right here, this is looking at the linear filters. This is in the feedback equals 0 model in blue. This is in orange, the feedback equals strength 1 Miles-Lisberger model. They're almost identical filters get fit in both cases. So both models agree that there does appear to be a change at this site. So far, so good. The Miles-Lisberger group would be very happy. But now we get into the difference. So now let's look at this site of plasticity. So if you assume that there's a feedback strength of 1 here, then basically the model only can fit if there is a LTP, a potentiation direction change at this site. If you assume there's no feedback and this isn't there, then you can only get the model to fit, and I'm not showing you that, but trust me, if there's LTD, if there's depression, functional depression at that synapse. And this really is what Mar-Albus-Ito and Miles and Lisberger said. So our model fits to the data, confirm these 2 different possibilities, but importantly say either one of them could work. Whereas people previously were saying no, either one can't, it can only be one or the other. Okay, so let's delve a little deeper. So now the key question is, how do these different synaptic changes lead to the same neural activity? How is it that these really both work? So now imagine what's happened is we've done gain-up training, so it's already been trained to increase its gain. We're now going to turn off the lights, and the animal's going to do VOR in the dark. And because it's been trained to gain up, its eyes are going to move more than it did previously. And what I told you previously is when you do that gain-up training, the activity of the Purkinje cell decreases with the increase in VOR gain. How does that occur in both models? This is really a review of what I said before. So in the simple Mar-Albus-Ito model, the no positive feedback model, that decrease in firing is driven by there being LTD of this pathway. In the strong positive feedback model, where we assume there's a strong positive feedback pathway, that decrease in firing is driven by there being LTP of the direct brainstem pathway, and then that having an inhibitory synapse onto the Purkinje cell that decreases the firing. And in fact, this neuron actually has LTP. Okay, so that's the first one. That's how the 2 different models produce the same output, but through very, very different sites of plasticity and very, very different mechanisms. Let's do one more. Now let's consider this motor cancellation task. This is the one where the animal's head moves together. So I should say the animal's already been trained to gain up. So this isn't a training paradigm, this is now a testing paradigm. So they've already been trained to increase their gain, but just to test, we're now going to have the animal move its head with vision, making it try to not move its eyes too much. And I should say this is what Miles and Lisberger used, and they did this because they thought, okay, this feedback pathway is now I'm intentionally making it hard to see. This is a motor output pathway. The animals aren't going to be moving their eyes very much. So they did this to try to isolate the head input to the Purkinje cells, while functionally, through a clever experimental behavioral trick, make this pathway relatively minimal because the eyes aren't moving much. The trick is, part of the reason they're not moving their eyes much is because they're making micro corrections all the time due to the external negative feedback pathway, and that's what Miles and Lisberger didn't consider as much. Okay, so again, you do this experiment. What you see is that after training, the activity increases with increase in VOR gain. And this is why Miles and Lisberger said, oh, that's direct evidence. We've removed the feedback pathway, and that's direct evidence that there was LTP at this site. That's what caused that increase in firing. Well, how does Mar, Albus, and Ito explain this? So Mar, Albus, and Ito, who said that there's LTD at this site, how can you get the increased firing? It's because the increased firing is due to the visual slip actually driving these neurons. So again, two very different mechanisms of getting increased firing. One has LTP here, one has LTD there, but one is using the visual, has a strong visual slip pathway. Okay. Finally, what I basically said is we can't separate out this old controversy. So what experiment might someone do to separate this out? So the kicker is in the Miles-Lisberger model, that feedback pathway of strength 1, what I didn't tell you is there's a double negative here. So it goes back to the first half of the talk. The Purkinje cells inhibit the brainstem nuclei. The brainstem nuclei inhibit some of the motor neurons, or actually a synapse up from the motor neurons, and then that sends excitation back. That's a positive feedback loop. In the Miles and Lisberger model, what I really meant by strong feedback was that this feedback was so strong in their model that it actually formed an integrator. If it forms an integrator, what you should be able to do is zap, electrically microstimulate the Purkinje cells, and you should see in response to that microstimulation that it's just like the saccadic input. You have a burst of input, and it gets integrated and maintains that activity. And that's actually what they said. They actually said this provides inertia to the system, and they said this might be useful for being able to view things through an occluder and keep your eyes moving. By contrast, in the Mar-Albus-Ito experiment, there's no feedback loop, so if you stimulate the Purkinje cells, you should cause a rapid change in eye velocity, and then it should decay right back. So in fact, Lisberger did this experiment in monkeys. He did a stimulation during this gray period and then stopped it, and it looks like the activity decayed back quite quickly. So it could be somewhere in the weak amplifying one, but it looks like that experiment suggests it's not in this strong feedback zone, that it's closer to the weak one. And this, I will now conclude, may help us resolve this controversy. So just stepping back to the big problem, studying learning in closed-loop tasks, conclusions about the sign, even the sign of plasticity, forget about the strength, but even the sign of plasticity depend critically on assumptions about internal circuit feedback and external feedback through the environment. Possible resolution to this 35-year-old VOR circuit controversy? Maybe there's LTP in the direct brainstem pathway. Both models agreed on that. And that's what Miles and Lisberger said. But you can still have LTD, which fits with a lot of the molecular studies in the Purkinje cell input pathway if appropriate visual feedback signals are present. And finally, altogether on both parts of the talk, breaking degeneracies between models is highly challenging, but may be addressable with causal perturbations in recordings. Okay, thank you. And this is just acknowledging the people who really did this work. But I know I'm running on time.
Yeah, so some people will need to leave shortly, but we can take a couple of questions while we're finishing up.
Thanks. Really like your approaches there. In the first part, you had an activation of the one side versus the other.
Right.
And I really like testing with lesions because I always say, like, you can figure out how it breaks. That's good. How acute was that lesion, and what if you had it over time and kind of looked at some more homeostatic plasticity?
Yeah, so that one was done with pharmacological inactivation. It was done two different ways, once with mucimol and once with lidocaine. And there you can look over time at recovery. Punchline in a lot, so I'm not sure you could recover from an entire half of the circuit being inactivated. More subtle ones will recover on the more like hour time scale, probably cerebellar driven. In the dark, there doesn't tend to be much recovery. There may be a little tiny bit. In the light, there does appear to be recovery, and it appears that that is actually cerebellar driven. So now we're actually trying to study how the integration is learned as another hook on a simple task for understanding how the cerebellum tunes a circuit. And now it's, in some sense, the simplest thing. How do you keep your eyes still? No movement stimuli. But that hopefully helps.
So the degeneracy in the second case, does that mean that, again, individual animals could solve it along a whole range of possible solutions that are mixed models of the two?
Yeah, so in principle, yes. And now we can just get philosophical about thinking about our guesses of how different animals solve it. My guess is that animals solve it the same way. But what seems to be interesting is actually that there seems to be a break between gain up and gain down. So Jennifer Raymond's lab has shown that it looks like gain up may be dependent on cerebellar cortex LTD, but not gain down. So this presents the possibility that actually it's even more paradigm specific about which mechanism. So it may be one mechanism is for one and one mechanism is for the other. And there's even subtleties when you get to sinusoidal frequencies about certain frequencies tend to be more dependent on cerebellar cortical LTD than others. So I think there are a lot of subtleties to work out. There are some other subtleties we could get into later, but I don't know. My guess is it's going to be, the usual thing of the mean is here and most animals are there and there's this much scatter, and I'm not sure whether the scatter would be enough to totally flip to two classes of animals solving the problem, but at least different paradigms it seems like might be solved, different ways like gain up versus gain down. And we don't have a good normative model of why that would be, although we have some thoughts.
All right, we should wrap up so that people can get on to their next thing. So let's thank Mark one more time.